presentation. Today, we have a good introduction to the Dr. Saosuke Imamura, and another distinguished presenter is from Professor Kumasude. Our two presenters will give us a very interesting topic that related to bioeconomy. And I think it's good time that we can start with the first presenter today. And I would like to introduce our invited presenter, that it's Dr. Saosuke Imamura. And during this time, you can share your slide, and all of presenter, you will have 25 minutes for present, and after that, we have Q&A about 5 minutes to share and give some suggestion. So I would like to check that Dr. Saosuke is ready. Okay, good. Good morning.
I share. Good morning. I
yes,
share
please.
my slide.
Yes, please. And first, I would like to briefly introduce about our keynote speaker. Dr. Saosuke Imamura received his PhD from Tokyo University of Agriculture and Technology in 2005. He worked as a JSPS research fellow at the University of Tokyo from 2005 to 2009. He joined Seoul University as assistant professor in 2009. And in 2012, he was appointed as assistant professor at Tokyo Institute of Technology. And in March 2021, he joined NTT Space, Environment and Energy Laboratories, Nippon Telephone and Telegraph Corporation. And Dr. Imamura had received a lot of rewards. He has received prestigious award from the Social of Genome Microbiology, Japan in 2010, and Nagasaki Science and Technology Foundation in 2017. His research interests include generation of the global environment, plant molecular biology, and biomass production. And today, it's time for Dr. Imamura to give us the presentation under the title of Regeneration of Global Environment Using Algae, Applying Molecular Mechanism of Carbon Storage Molecule and Their Applications. So I would like to give this valuable time for Dr. Imamura. Please give us the informative presentation.
Okay, thank you very much for the kind introduction. First of all, I want to express thanks to the organizers to give me a good chance to present our recent research. I'm Saosuke Imamura from NTT Space, Environment and Energy Laboratories, which was established about 1 year ago. Before that, I was working at Tokyo Tech and study about the molecular mechanism of biomass production. And most of today's my talk will be done at Tokyo Tech. Okay, this slide indicating today's my talk's content. After a brief introduction, I want to talk about switch protein for triacylglycerol tag accumulation. Next, I will talk about improvement tag productivity by molecular genetic approach. After that, I want to present utilization about algal starch and its accumulation mechanism. And finally, I want to summarize my talk. Okay, let me start with the introduction. And this slide indicating the CO2 emission and also absorption on the Earth. As you can see, the absorption of CO2 by soil and ocean is large. So focusing on ocean is a good strategy to reduce CO2 in the atmosphere. To reduce the excess CO2 on the Earth, especially in the ocean, the ability and characteristic of microalgae seem to be excellent because They produce high biomass productivity, no requirement of arable land, and so on, shown here. So, studying about biomass production of microalgae will give us important information to solve several issues that occurred on the Earth. Today's my talk is about oil and carbohydrate. First, I want to start with the oil story. Generally, microalgae accumulates a neutral lipid in the cells and several stress conditions, such as nitrogen depletion. As you can see, body stained neutral lipids were increased after exposure to nitrogen depletion conditions. The main lipids in the cells are triacylglycerol's tag. And this tag is a good feedstock for biofuel production. So far, large-scale and commercial biofuel production has not been established. I think there are two major points to be solved in the field of fundamental research. One is underlying molecular mechanism, how microalgae regulates the accumulation. To increase the oil content in the cells, important step for biofuel production using microalgae. But it is difficult to increase the oil content without the basic molecular mechanism. The other point is this kind of trade-off relationship between oil production and cell growth because, as mentioned, tag is generally accumulated under the stress condition, such as nitrogen depletion. So, to improve the tag productivity, we have to achieve high level of tag and normal growth speed at the same time. I think today's my talk might answer these two important points, I think. Next, I want to introduce our main material using in this study. We are using the unicellular lethargy cyanidium Cizon melora. We are calling Cizon. And Cizon has several features that are indicating in this slide. Among them, I want to emphasize the following 3 points. First, the complete genome sequence has been already determined. Second, targeted gene disruption by homologous recombination is available. And finally, the ability to grow in the ocean. So Cizon is thought to be one of the good model organisms to understand photosynthesizing eukaryote due to several these algae's features. Okay, now I want to move to the second part. TOR is a switch protein for tag accumulation. Okay, this slide is indicating what I want to explain in this section. Using the Cizon in 2015, we found a switch protein TOR, T-O-R. This function is similar the switch located on the wall. If we want to turn on the light, we will change the switch from off to on. So this towards protein's function determine tag accumulation or not. The TOR is abbreviation of target of rapamycin and well-conserved protein kinase among eukaryotes. And this TOR kinase activity can be inhibited by its inhibitor rapamycin. So this protein is called as target of rapamycin, TOR. And this slide indicates results showing TOR is a switch protein for tag accumulation. Because as you can see, the body piece stained neutral lipid were accumulated 24 hours after the rapamycin treatment and further accumulated at 48 hours. And quantification data indicated that tag content was increased about 9-fold compared with the control condition. So these results clearly indicated that TOR inactivation lead to tag accumulation. The tag content under the rapamycin treatment condition was almost comparable that's under the nitrogen depletion condition. So we can say that TOR signaling is a major pathway that regulates tag accumulation under the nitrogen depletion condition. Now, I'd like to summarize the main point of this section. Under the nitrogen replete condition, a TOR activity is active and play important role on the cell growth. In contrast, under the nitrogen depletion condition and also rapamycin treatment condition, TOR kinase activity is inactive, and this inactivation resulted in tag accumulation. So the TOR activity determines the cell growth or tag accumulation. Namely, we can say that TOR is switch protein for tag accumulation in microalgae. Okay, now I want to move to the third part, the improvement of tag productivity by molecular genetic approach. The finding of the switch protein TOR is important to understand the regulation of tag accumulation, because we can induce a tag accumulation anytime by just rapamycin addition to the algal culture. But the TOR inactivation is not good condition for tag production from commercial point of view, because as mentioned in the previous slide, TOR plays an important role on cell growth. So if we want to increase the tag productivity, we have to achieve concurrently high tag content and normal growth speed under the normal growth conditions. To achieve these things, we next have to review how TOR regulates the tag accumulation. Namely, we have to identify this kind of regulator shown by question mark. So as a first step to get information for the identification of the regulator, we perform genome-wide transcriptome analysis using DNA microarray. This table showing the genes of which products are involved in fatty acid and tag synthesis with expression ratio under the TOR inactivation condition and nitrogen depletion conditions. Genes highlighted by red mean those transcripts were increased under the both conditions. I will call this gene and GPAT1 and the other is GPAT2 hereafter, because both genes are encoding glycerol 3-phosphate acetyltransferase, GPAT. GPAT catalyzes the first step of de novo tag synthesis in the ER, or catalyze the first step of de novo galactolipid synthesis in the chloroplast. So we first check the localization of GPAT1 and GPAT2 in citizen cells. This time I will not show the localization result, but the result indicated that both GPAT1 and GPAT2 localized in the ER. So we can easily imagine that the two proteins are involved in the tag synthesis in the ER. However, some previous paper indicated that reaction catalyzed by the DGAT, the final step of tag synthesis, is rate limiting step in the tag synthesis. On the other hand, our result lies a possibility that GPAT1 or GPAT2 expression is important in the tag synthesis. To check our scenario, we overexpress GPAT1 or GPAT2 in the cells and observe the tag accumulation in each strain. The upper panel indicating the body piece stained cell, you can see the very clear and intense lipid droplets were observed in GP1, GPAT1 overexpressor, even under the nitrogen replete condition. But this kind of accumulation was not observed in GPC and GP2. Under the same condition, the tag content in GP1 was significantly increased by about 19-fold compared with GPC. This 2% dry weight of tag is larger than that under the nitrogen depletion condition. So this result clearly indicates that the reaction catalyzed by GPAT1 is a main rate limiting step in season cells. Well, this high tag content seems to be very good, but how about this strain's growth speed? Surprisingly, the growth of GPAT1 overexpressor was comparable that of the control strain GPC. So we next measure the tag productivity at each growth phase, mid exponential, late exponential, and after exponential phases. As you can see, the tag productivities were drastically increased with incubation time. This will coincide with the result. GP1 is showing high tag content and no growth inhibition. The tag productivity was greatest during the after exponential phase. The ratio was more than 56-fold increase compared with the control condition. So we succeeded in a high level of tag and a high growth speed at the same time. Namely, we can say that we solved the most critical problem to be solved in the field of tag production in microalgae. Next, we interested in transcription factors. As you know, each transcription factor, TF, generally regulates set of genes of which functions are interconnected. So driving the overexpression of transcription factors that control the expression of GPAT1 could be an efficient method for enhancing tag accumulation. So we next try to identify the transcription factor that involve in GPAT1 transcription. Using the two accumulation condition, tag accumulation conditions, -N and tau inactivation, we check the available transcriptome and proteome data and picked up candidate of 13 transcription factors. Then we constructed each transcription factor overexpression strain and checked the tag accumulation using body stain method. As you can see, but it is difficult to see the neutral lipid, but we identified a lipid droplet in 4 transcription factors overexpression strains, even under the plus N condition, namely normal growth condition. Next, we check the level of transcription in each strain and reveal that only LPAT1 transcripts were increased in all 4 strains. Unfortunately, GPAT1 transcript was not increased in overexpression strains. But we further analyze LPAT1 on tag accumulation. Our immunostrain analysis revealed that LPAT1 localized in ER, and it is known that this protein catalyzes a second step of tag synthesis in the ER. So when we overexpress LPAT1 in the cell, we observe the increase of the tag level in comparison with that in the control strain. This indicates that LPAT1 is one of the rate limiting steps of tag synthesis as well as GPAT1. Okay, now I'd like to summarize shortly GPAT1 and LPAT1 stories in this slide. Under the -10 and the tau inactivation condition, the transcription of GPAT1 and LPAT1 are increased. In the case of LPAT1, the transcription is regulated at least 4 transcription factors, BRD1, MIB3, HSF1, and MIB4. So far, we have no information about TF that regulates the GPAT1 transcription. The contribution of GPAT1 and LPAT1 are extremely different since induction ratio by GPAT1 overexpression was so high compared with LPAT1 overexpression. Namely, the step catalyzed by GPAT1 is a major rate limiting step for tag synthesis in this algae. We recently found further regulators for tag accumulation. The regulator is FAX1. This is encoding fatty acid transporter. We thought that we can increase the tag content by enhancing fatty acid transport from chloroplast to ER, because the fatty acid used for tag synthesis are synthesized in the chloroplast. Actually, this FAX1 localized on the outer envelope of the chloroplast. This line indicates the tag content and its fatty acid composition, the tag in the FAX1 overexpression stream. As you can see, the tag content was increased by FAX1 overexpression. With respect to the fatty acid composition in the tag, the most interesting point is the overexpression stream is containing the short fatty acid such as 14,0 and 14,1 fatty acid. These fatty acids are suitable for jet fuel for airplanes, so we assume that further investigation of FAX1 will be useful information not only to control the tag content, but also its fatty acid composition in the tag. As mentioned in the previous slide, we identified several regulators for tag accumulation, but our analysis until now is to overexpress only one protein in the cell. If we can enhance each function in one cell, there is a chance to increase drastically tag content, also its fatty acid composition. So as a next step, we want to try to construct superalgae of which tag contents are extremely high through expressing these proteins in one stream. Next, I want to move to the starch stories. Because as you know, algal biomass means algal cell itself. And oil is only part of the algal biomass. So we have to consider utilization of the oil algae residues. For example, the residue is containing a large amount of starch. So if we can produce useful chemical using algal residues, we can reduce the oil production cost since there are chance to get a large gains. In 2017, we succeeded in establishing one system in which we can convert the oil algal residues, especially starch, into alkyl liberinate or alkyl lactate using chemical catalyst. Interestingly, we can selectively produce each chemicals using different catalysts. Alkyl liberinate can be used for pharmaceutical and plasticizers, and alkyl lactate can be used for biodegradable polyesters. And this study was done with my colleague. They are measured these chemicals, but I'm a biologist, so what I can do next is to increase starch content in the cells. And recently, we revealed that the switch protein TOR is also involved in starch accumulation. As you can see, the starch content will clearly increase after Rapamycin treatment. Recently, we also revealed that how TOR regulates the starch content, namely identify this regulator shown by question mark. Right? And TOR is a protein kinase, so we thought that the phosphorylation status of starch-related enzymes under the TOR inactivation condition and the change resulted in starch accumulation. So to check the scenario, we performed a phosphoproteome analysis before and after the Rapamycin treatment. And this graph indicates the proteins of which phosphorylation status was decreased or increased after the Rapamycin treatment. Among them, we found two proteins that's related to starch synthesis, and focusing on the following two proteins. One is glycogen 1, GLG1. The other is glycogen synthase, GS. I want to explain the function of GLG1 and GS during the starch synthesis. And I think you can easily understand the both proteins function when you imagine the PCR reaction. Because the GLG1 and GS functions are similar to DNA primer and DNA polymerase, respectively. So GLG1 is necessary to de novo synthesis, de novo starch synthesis, and GS is necessary to elongation the starch chain. Okay, the next to check the both functions in the starch synthesis in season cells, we first constructed each overexpression strain. As you can see, the starch content will clearly increase in the GLG1 overexpression, but not in GS overexpression strain. So we next further investigated GLG1 function on starch accumulation. Because the serine 613 was detected as a phosphorylation site by TOR in the LC mass analysis, we changed the serine 613 to aspartic acid to mimic the phosphorylated GLG1, or change to alanine to mimic the phosphorylated GLG1. Interestingly, the increase in starch was significantly reduced in the phosphorylated GLG1 mimicking overexpression strain, but not in the phosphorylated GLG1 overexpression strain. This clearly indicates that the phosphorylation status of serine 613 is regulated by TOR, and its status determines starch accumulation or not. Namely, under the normal growth condition, GLG1 is phosphorylated by TOR signaling pathway and inhibits the de novo starch synthesis. In contrast, under the TOR inactivation and also minus N condition, the amount of phosphorylated GLG1 is reduced because TOR kinase activity will be inhibited. Then it triggers starch accumulation in the cells. Okay, now I want to move to the final parts summary. Okay, I'd like to finish by making the following 6 points shown here. The first point is TOR is a switch protein for tag and starch accumulation in microalgae. And the reaction catalyzed by the ER localized GPAT1 and LPAT1 are the rate limiting step for the tag synthesis in season cells. But the contribution of two proteins to the tag accumulation are extremely different. Namely, the GPAT1 is a major rate limiting step in these algal cells. LPAT1 is regulated by at least 4 transcription factors in response to the tag accumulation condition, such as -N and TOR inactivation condition. These regulators, including the FAX1, GPAT1, LPAT1, and also 4 transcription factors, would be a potential target for improvement for tag productivity in microalgae. And the algal starch is regulated by GLG1, and also its phosphorylation status is very important for the starch accumulation and variable feedstock for useful chemical production. And this point is very important and message from me, and TOR is a good target to understand biomass production in microalgae. And I believe this kind of basic research and bench scale demonstration, how we can improve the tag and starch productivity, will help to establish a commercial biofuel production using microalgae. And I want to again emphasize that TOR is a good target for the purpose. Okay, finally, I want to thank the people who have been involved in this study and also financial support. And that's all I have to say, and thank you very much for your kind attention.
Thank you for your very nice presentation. And thank you again for the inspiring presentation, your depth of the understanding for how to produce a very super algae. And it's the most promising algae in the futures. And it's a very interest to know that we have some switch to turn off like the light that it's good chance to accumulate a very view for the futures. I think it's not easy that we can looking for a light switch to turn on the good direction for the opening the light, as you try to present us in the very informative information. And if any question and comments, it's welcome for everyone. It's a good chance to sharing your knowledge and information. You can post your question from the chat, or you can raise up your hand, and then you can asking our speaker here. Yes, Dr. Sudet, have some question and comment, please.
Yeah, thank you very much. Very interesting and informative talk from Dr. Imamura. I have two questions. One is, what is the fatty acid composition of the tag that you normally get? And whether that fatty acid composition is suitable for producing biofuel. The second question is, I think all these super algae strains are all genetically modified. However, these genetically modified algae have to be cultivated in large scale in open system, in open pond systems. So how do you think the society or the government will give approval for using genetically modified organisms in large scale? Thank you.
Thank you very much. The first question is maybe the fatty acid composition in the tag, containing tag. The tag is containing the 3 fatty acid, and this fatty acid composition is varied and depend on the organism. But the algae and also plants generally containing the fatty acid carbon length is 16 and 18. And 16 and 18 is suitable for biodiesel. And more short fatty acids such as 14 and 12 and 10 is suitable for jet fuel. Did I answer the first question?
Yes, yes, thank you.
And the second question is, yes, we have to consider the GM. And as I mentioned in the introduction of my material, Cyanidum season melora, and I mentioned that this algae, we can apply the targeted gene disruption by homologous recombination. So, and we recently developed one system that we can repeats utilize a marker to select a GM. But after that, we developed a technology to delete the selection marker. It means after deletes the selection marker, the organism is not GM. So after constructing the super algae through overexpression by each target gene, we can create the organism without a GM.
I see. Okay. Thank you.
Thank you very much.
Question?
Yes, please.
Did you hear me? So Langsima Shoku from ASEAN University. Okay, so your topic is very interesting, and I think it's a very new topic for me. So could you please give in the future, is it possible to do like a large scale as Dr. Sudet asked you, or just this still be in the pilot scale, or just in the laboratory scale? Is possible that in the commercial scale or in the large scale, how how about the percent you to extract for the any that fatty acid to be like the biodiesel or biojet?
Sorry, it is hard to hear.
Okay.
Okay,
catch up there.
I will transfer the question again. That it's mean from the laboratory, and do you have the more pilot scale or some commercial
I
scale?
see.
Do you have the yield or the productivities, not
Thank
only
you.
from lab?
Yeah. Unfortunately, we just check the productivity in very small scale, not the commercial level. So I want to try to such kind of thing. Yeah, that's a future subject to be solved and checked.
Thank you.
Sorry.
Yes, because it's very interesting topic, especially we can success for the laboratory. And if we can see from the pilot scale and also from the sea water in really life, that will be very interesting, because I think that will be a very promising, even in this time, if we can do a very high production and make more a super algae that you really try to driving this platform. I think it's very, very important for our world to be like a future source for the energy storage. And I think we still have maybe some quick question if someone would like to share a suggestion.
If there is time, maybe I can.
Okay.
Yeah, there is a lot of interesting questions. For
just a quick-
example, the growth of algae, one of the main limitation is light availability.
Mhm.
So how do you think, we can achieve this efficient growth, even though the light limitation is there? Do you have any ideas?
Yeah, one of the ideas is we have to reduce pigment content. And we recently very interesting strains of which pigment content is very low. So the light intensity, light efficiency to the bottom of the culture tank easily reach when we cultivating such kind of strain. So such kind of strains may solve such kind of issues.
Yeah, it's a good idea. Yes.
Yeah, if I have a chance to the next conference, I want to introduce you the super algae.
Okay.
Yeah, thank you
Thank
very
you.
much.
Okay. Okay, thank you for the fruitful discussion. And again, I would like to thank you, Professor Imamura, to give us a very good presentation. And the next invited speakers of today is Professor Sudet. And you can sharing your slide now. And first, I want to introduce Professor Kumar Sudet. He is the professor at the School of Biological Science, University, ASEAN, Malaysia, and is well known as his research on bio-based and biodegradable plastic produced by micro, especially PHA. His research activities are found by several international, national, and university grants. And a lot of students nowadays study with Professor Sudet. More than 30 master students and 38 PhD students has graduated under his supervision. With more than 200 scientific publications and 8 grant patents, he is among the top research scientists in Malaysia. He is also the chief editor of Malaysian Journal of Microbiology, a fellow of Academy of Science Malaysia. So today it's very good chance for ABB that have Dr. Sudet with us. And Professor will give us under the title of Bio-based and Biodegradable Plastic from Renewable Resources and Sustainability Process. So now it's your time.
Okay, thank you. Thank you very much, Dr. Patra. And also thank you to the organizing committee, especially Dr. Pilani from KAPI. Actually, we in USM, myself and Dr. Pilani have been having collaborations since 2016. So it's almost 5 years we have known each other. And I think I have also presented at previous ABB, and I see some familiar faces. So it's nice to see all of you all again. Yes, I think we all agree that we are facing increasing challenges in our daily life. So we need to think of sustainable processes for our future. An earlier talk on the energy is one of the important requirements for human civilization and also for all kinds of general well-being. And I think besides energy, materials is also another essential requirement for human. So these are two challenging topics, simply because in order to make them economically viable, cost is the major issue. And I think we all agree that for successful and also sustainable bioeconomy, we have to reduce the cost. So my talk will be focusing more on how are we trying to reduce the cost of producing bioplastics. So my university, I'm not sure if many of you all know University Science Malaysia. This is a university that is located in the northern part of Peninsular Malaysia, very close to the Thai border. So the university is on the island, Penang Island. I think many Thais come to Penang for holiday. Of course, now because of the pandemic, many people can't travel. I hope soon this will be possible and we can visit each other. And University Science Malaysia have been very passionate about sustainability since very long time ago. And we have many academic programs, and also we try to develop a university that is focused on sustainability. And we are very fortunate that according to the Times Higher Education ranking recently for 2021, University Science Malaysia is ranked 39th in the world for sustainability. And in Asia, in Asian countries, USM is number 2, and number 1 is Chulalongkorn University in Thailand. So this is very nice, and we hope we can collaborate and work more on these sustainability aspects. And like energy and getting our energy resources has been talked earlier by Dr. Imamura. So I'll be focusing more on material. And when it comes to material, I think we all know that plastics is one of the most widely used material. We cannot escape, we cannot run away from using plastics. I think if you look around you, there will be many items made out of plastics. So here in this slide, you can see some of the common plastics that are widely used. And what sometimes we don't realize is that the clothes that we wear are also either made of plastics or contain significant quantity or component of plastics. So like in these two labels that you can see, like polyester and rayon or polyurethane and acryl, these are all plastics. So sometimes we don't realize that when we talk about plastics and we want to reduce plastics, it is not so simple. I think this is what we have to remember. And plastics, I think they are very excellent material. I don't think we would be able to enjoy this modern lifestyle without plastics. So plastics are good. I think plastics are excellent material. The problem is after use. Even like our clothes, when we wash our clothes after use, thousands or millions of microfibers are released from our clothes. So all this ends up in the ocean. So these are the problems that we are facing. So what can we do? So microplastics is one of the issue. Other than microplastics, I think we all agree that the commodity plastics, especially the single-use plastics, are also causing a lot of problems. Sometimes we don't see it in front of us, but I think we all know that plastic waste is a growing concern for everybody. And this is a problem that crosses borders, and it is like problem that is faced by all countries. So this kind of issues requires that we need to come up with either more efficient plastic disposal processes and also treatment processes, and at the same time those plastics, especially single-use plastics that are biodegradable, would be advantageous. So because of these different types of solutions, there are many different approaches that are being taken by various countries. So bio-based material and biodegradable materials, they all sound technically almost the same, but their properties are different and their ability to degrade in the environment is also different. So this causes confusion also. So I think a lot of information and education is necessary to understand the different types of plastics and also the different purposes why different types of plastics are being produced. So there are some logos like this which are already in use. Like in Japan, you have green pla, and in Finland, you have this nice, cute Apple logo, which is to show that these are all addressing some environmental issues. And you also have compostable plastics and also like those which are biodegradable. In Thailand, this is from a conference that I attended just before the pandemic. I think it was in 2019 in Bangkok. There are a lot of efforts to produce various kinds of bioplastics. And here are some pictures, plastics that are produced from starch. You have modified thermoplastic starch, and then you can also produce polylactic acid. The starting feedstock for making polylactic acid is starch. So these are all some types of bioplastics, which are not necessarily biodegradable, but they are plastics that do not rely on petrochemical resources. So these are also considered efforts and initiatives which are necessary. And in my lab, what I have been focusing on is on producing polyester that is 100% biodegradable. So these are a kind of polymer, a high molecular weight polymer that is produced by many kinds of microorganisms, mostly from the prokaryote and archaea classes. And these polyesters are known to be biodegradable. And the key enzyme that is involved in the polymerization of this polyester, which is called polyhydroxyalkanoate, or PHA in short, is called PHA synthase gene. So a lot of studies has been done to identify synthase genes which are can be expressed highly, and also the enzyme which is very efficient in polymerizing and which can polymerize a broad substrate range, which will be necessary to produce different types of bioplastics. And in Malaysia and also in Thailand, I think one of the major agro-industry product is like palm oil. Malaysia is currently the second largest producer of palm oil in the world, behind Indonesia. And we have large quantities of waste products from the milling process of palm oil. So these are all renewable and also interesting feedstock that can be used to feed microorganisms to produce PHA. And one of the challenges in using PHA is the recovery of the PHA from the bacterial cells. So these are polymers, just like triacyl glycerols that are produced by algae. These are all compounds that are accumulated inside the algae or bacterial cells. So the challenge is, how do we get it out of the microorganism? So the recovery and processing is a major issue, and it costs almost half of the production cost. So these are all issues that we have to deal with in order to reduce the cost of bioplastics. But the material itself, once you have extracted and purified the polymer, it is 100% biodegradable, as is shown here in this slide. So if you leave a PHA film on the soil in a tropical climate like in Thailand or in Malaysia, it will degrade within a period of about 1 month, as you can see in this slide. And PHA is also a thermoplastic, which means that you can mold and you can process it, just like how you process petrochemical plastics. And it is biocompatible. You can also make medical devices and implant it into human body, and our body will not reject these PHA-based medical devices. And it is also renewable. So we can produce PHA from plant-derived resources. So like I mentioned just now, palm oil is one of the major renewable resource that we can produce in large quantities. And currently there are already a lot of applications for palm oil. Of course, food as an edible oil is the major application of palm oil. Secondly, it is for oleochemicals. A lot of oleochemicals, different types of fatty acids are all purified from palm oil. And also there has been a lot of effort to produce biofuel, like biodiesel from palm oil. And what I have been doing for the past 20, 30 years is to focus on producing biodegradable plastics. And the oil that you can get from palm oil, there are two different types of oils. You can actually get palm oil from the mesocarp of the palm fruit, and you can also get the palm kernel oil. So they both have different fatty acid compositions. So these are both useful types of fatty acids for the production of biodegradable plastics. And what is the most attractive about palm oil is the productivity. So as you can see here in this graph, which compares the productivity of all vegetable oil, and you can see palm oil stands out as the most productive. So you can produce more than 3 tons of oil from a hectare of land in a year. And this is far more superior than any other types of vegetable oil. So this is what we all aim to do as microbiologists or biotechnologists. We are all trying to improve the efficiency of producing whatever compounds that we are interested in. So here you can see we already have a very efficient crop which can be used to produce oil. So the process of getting the palm oil involves various milling process. So the fruit has to be crushed and squeezed in order to obtain the crude palm oil, as can be seen here. And the crude palm oil is orange in color and very fruity in smell. And during this process, of course, a lot of oil is leached from the mills, and this oil becomes interesting and also kind of like a feedstock that we can work on to produce various kinds of value-added products. So here I've shown one example. We can use the sludge palm oil, which is wasted from the milling process, and it can be fed to genetically modified microorganisms. The microorganisms that I've been using here is Cupriavedus necata. This strain has been metabolically engineered to use oil efficiently to produce the kind of PHA that has got high commercial value. So we are developing various optimum fermentation processes to produce and to convert this waste oil into PHA. So the other resource besides oil from the palm oil industry is the sugars that you can also get from the oil palm trunk. So this has been neglected for many, many decades, simply because not many have looked into the oil palm trunk, because the fruit is already producing very profitable oil. So nobody bothered about the trunk. But the trunk is another valuable resource. So normally the trunk is wasted during replantation of the oil palm plantation. So oil palm is replanted every 25 years. And this is because after 25 years, the oil productivity starts to decrease. So the normal practice is all the old trees are cut, they are felled, and then they are chipped into smaller pieces and left in the plantation. So they are not used for any applications. But we found that together with some research collaboration with Japanese researchers, that the trunk contain high amount of liquid, which is rich in sugar. So this bottle that you see on top here, which contains like a sugar cane, is actually the sap that you get from the oil palm trunk. So the oil palm trunk can contain up to 80% of its biomass, this liquid, which is rich in sugars. And it is not only sugars, it also contains other growth factors and vitamins and minerals that are necessary for microbial fermentation or for microbial growth medium. So we have found that this is a very valuable resource that can be used for various fermentation processes. And this project now is funded by both Japanese government and also the Malaysian government through SATRAPS project. I think some of you all may know SATRAPS. This is a project funded by JST and JICA, and which is for the science and technology research partnership for sustainable development. So this was just started during the pandemic in 2019, and the project is ongoing. So there are many institutes from Japan and Malaysia which are involved in this project, and also companies from Japan and Malaysia which are involved in order to commercialize this this oil palm trunk, and to add value to this oil palm trunk that has been neglected for many years. So this project is going very nicely, and we have actually established various processes to use the oil palm trunk. So this is what normally is done to the old trunks. So during replanting of the oil palm trees, the old trees are cut and left in the plantation. And as you can see here, this is all rich biomass that are wasted. It can be used for high value product application. And also these old oil palm trunks, which are rich in sugars, if you leave it in the plantation, it will cause the growth of various fungus, as you can see here in this trunk. And this fungus can be pathogenic to the oil palm trees, and these are not good practice. But it has been done like this for many years, because to remove the old oil palm trunks from the plantation is costly. And in order to reduce the cost, normally the trunks are left in the plantation. So what we are doing is we are trying to add value. We are trying to develop new processes and also products from the oil palm trunk, so that the cost of removing the trunk from the plantation can be met by developing high value products. So these are examples of the oil palm trunks that you can get in very large quantities, and you can get tons, millions of tons of oil palm trunk throughout the year from various parts of in Malaysia, in Peninsular Malaysia, and also in the Borneo Island, like in Sabah and Sarawak. And this can be developed into some products. So I'll be showing some of the products that we are able to produce currently. So we have established a pilot plant to process the old oil palm trunks And currently, the main product is to produce fuel pellets. So this kind of fuel pellets are entirely made from the oil palm trunk. And during this process, the trunks are squeezed, and by squeezing, a lot of the sap is extracted, as you can see in the middle of this slide. And this sap is rich in sugars. So which means you can also develop processes to produce algae or bacteria or any kinds of microorganisms in large quantities for various products. And we are focusing on producing biodegradable plastics. And this whole pilot plant is designed by adapting the concept of circular processes, whereby no waste will be released into the environment. So this plant is equipped with an IC reactor to anaerobically treat the waste and to produce methane. So the methane is then used as a fuel to heat up and also to provide electricity for some of the processes. So this is the main objective of this SATREPS project, and we are very confident that it will be successful in producing bio products and also establishing a new bioeconomy. And in order to produce biodegradable plastics, what we are doing is we are scaling up the process. So from the laboratory scale, we have now gone into 1,000-liter scale, and we are trying to come up with processes to produce the kind of plastics that we want. So PHA, I'm not going to go into more detail on PHA. There are many kinds of PHA that can be produced. And one of the commercially attractive type of PHA is PHBHX, which is written here, as P3HB Co3HHX. And by varying the monomer compositions from 10% to 30%, you can get materials which are hard and crystalline. And by increasing one of the monomers, you can make the polymer very flexible and elastic. So these are all can be controlled by using different types of strains and also by controlling the feeding of the feedstock. So for this, of course, we use genetically modified microorganisms. So the next challenge is how do we recover the resulting polymer, which I mentioned earlier, which is inside the bacterial cell. So this is also another challenging process. Normally, the bacteria, after it accumulates the polymer, it has to be hydrolyzed and the polymer has to be released from the cell cytoplasm. And these polymer granules are very tiny. They measure only about 0.5 micron in size. So these are smaller than the bacterial cell. So you can imagine how difficult it would be to separate these tiny polymer granules from the bacterial cells. And normally this process involves solvent extraction, chemical digestion by using enzymes and chemicals, or the combinations of enzymes and chemicals. So all these require a lot of expertise and also cost. And the process, if it is not well controlled, it will affect the molecular weight of the polymer. So this is also another challenging task because you want to maintain the molecular weight as high as possible. And by adapting this kind of processes, we cannot avoid or run away from the waste treatment problem, because at the end of the purification process, you still have the residual cell biomass which has to be treated. So all this adds to the cost of producing the polymer. And coincidentally, about 10 years ago, we accidentally found that the bacterial cells containing the polymer can be fed to animals. And the first discovery was when we found that the cells were eaten by rats. And the rats, after eating the cells, their feces contains only the polymer. So the polymer is not digested, and the polymers are secreted out as a fecal pellet. So from that, we got this idea that we can use animals to purify the polymer, and not use chemicals or solvents. So here we have tested many different kinds of animals, and we found that mealworms are quite interesting. So this picture here shows mealworms eating the bacterial cells. So the lump of brown colored thing that you see in the middle is the bacterial cell that has been dried. And the mealworms are eating these bacterial cells, and the white granules that you see around them are the fecal pellets. And these fecal pellets, you can see here in the scanning electron micrograph, they are basically all the polymer granules. And this principle is very, very simple to understand, because the polymers are not digested by the animals, so they are released. And the animals, they absorb the nutrients like proteins and lipids, but the polymers are released. So this process has now been scaled up by one of my PhD students, Dr. Zainab, and we are now able to produce PHA in large quantities in the lab. So I'm trying to rush through these two concepts of producing efficiently the PHA from waste, and also to recover the polymer in a cost-effective process. So in conclusion, what I've attempted to show is that bio-based and biodegradable plastics are necessary to replace some, not all plastics, but at least some commodity plastics, especially the single-use plastics. And cost reduction is necessary. So this is where innovation is required to establish sustainable processes that are economically feasible. And the palm oil industry, of course, in Malaysia, I'm focusing on palm oil, but in other countries, there can be other industries that can provide both oil and sugars as a feedstock for bioplastic production. And these concepts can be summarized as industrial symbiosis and circular economy, which I think will be the way forward to reduce costs and also to protect our environment. And of course, there are many issues that we also have to keep in mind. For example, loss of tropical rainforests and biodiversity, wildlife, the habitat, global warming, climate change, and conversion of food to fuel. These are all serious issues that we have to keep in mind while developing new sustainable processes that would be more eco-friendly. So with that, I thank you, and I will be happy to answer any questions that you have. Thank you very much.
Thank you very much, Professor Sudet, for very valuable information. And I think we learned a lot from your presentation, and it will be very wow for me to know that every part from the farm, we can make it like a very high valuable. And it's not only from the small scale, now you can develop, and also the company see this valuable, and he tried to drive the technology to be like a sustainable. So also it's come into the bio circular and green economy that it's very famous, and everybody know that this topic is very hot issue. So if anybody would like to share or asking some questions, so it's time for you to give some question or comment.
It's before lunch, so everybody will be hungry.
Maybe, and I think it's a good time before we break to discuss about more very informative before we digest to the lunch. Okay, and may I start for the simple question, because for the bioplastic synthesis that you would like to develop, and it seems accumulation into the bacteria or its secret outside. I would like to know the production of PHA that on your process.
So PHA is an intracellular product. So it is accumulated in the bacterial cell cytoplasm. So therefore, the challenge is how we can break the cells and extract the polymer from the bacterial cells. Of course, if there can be new innovation which can secrete the polymer naturally from the bacterial cell, that would be more effective. But currently, there has been no such, it is not possible to secrete the polymer from the bacterial cell. Yep.
It's still some limitation that we
Yes.
need to overcome.
Yes.
Okay, and we have one more question. Would you suggest that the cost for this system is compatible with the present industry or not?
Yeah, that's the question that we are all trying to answer. So that is also why I'm trying to find alternative approaches, and I think one of the key what you call idea that we can use is how to develop a biorefinery concept in order to reduce the cost. So the concept of biorefinery is just like chemical refinery, where every component are used for something. So instead of having a linear process, for example, to produce bioplastic, you start with a feedstock and you produce only one product. I think this will be too costly. So this is what we have seen. So in order to reduce the cost, I think we have to produce more things from the same process. And this was, I think, similar to what Dr. Imamura also mentioned, not only to produce a high starch or high triacylglycerides, but also some other chemicals from the process. So that is exactly what we are also trying to do. So the mealworm recovery process is interesting because the mealworm becomes another product, because mealworm is a high value product which currently is being developed as a future source of protein for not only animal feed but also for human food. And I think in Thailand, in Thailand, I think you are very already you consume insects. It's normal. It's not something that psychologically people will feel difficult like in other countries. But definitely, I think in the future, the supply of meat is going to be a problem. So we have to look at alternative sources of protein. So mealworm is attractive. So if we can combine mealworm production with PHA recovery, I think that is one way how we can reduce the cost and minimize waste.
Yes, I agree with you that we need to looking for another protein sources also. And it seems like you try to do like a zero waste process. Every byproduct, it's not, it can make itself valuable. So it seems like a better for biocircular economy. And it means everything it's a benefit. So yeah, that is very innovative, and I think it's many jigsaw that we need to connect to make this platform it's useful in a daily life, and maybe many factory can apply your technology to apply it in the futures. Yeah, very interesting. And do you have any question and comment from the floor?
I think Dr. Rungsima.
Yes, please.
Yes, Sima from ASEAN University. Yes, thank you for your nice presentation and interesting information. So as you know, we work together to apply application for PHA. But I would like to know about you try to use another variety of worm, maybe another insect to try. Is it different or not that you extract or purify the PHA from the
Yes. Yeah, of course. I think that would be something that we also can do to look for other more effective insects or animals that can recover the polymer efficiently. So this is something like like coffee luwak. I think coffee luwak also follows the same concept. I think in Thailand they use elephant feed coffee beans to elephants, and then the coffee bean is recovered from the the elephant's feces. And in Indonesia they use a kind of a wild cat civet to eat the coffee fruit, and then the beans are recovered. So these are all very exotic coffee. So the concept is almost the same, where we feed the bacterial cells to animals, and then the animals cannot digest the polymer, so it comes out in the feces. So this, like you said, there may be more suitable animals or insects. We need to look for it. So I hope in Thailand also you try to look for better, more improved processes. Yeah.
As you know, we and Dr. Plani work together with the silkworm.
Yeah,
Maybe
silkworm.
it's,
Okay.
yeah, interesting to use to try
Yeah.
this silkworm, not
Yes.
only the mulberry silk, also another UV silk that they eat the cassava leaf for feed.
Yeah.
Okay. Thank you very much.
Yeah.
So
Thank you.
maybe in next work together in the
Yes.
future.
Yes.
Thank you.
Thank you. And just one more quick question about the mealworm system. It is very biological and environmental friendly, but however, would it be acceptable in accepted of some ethic? That is just for quick.
Yeah, I don't know. I don't know, because again, this is something, we sometimes when we develop this kind of thing, it is difficult because this involves a society, culture, so the easiest way to get acceptance is if you feed the mealworm to animals as a source of protein, like for aquaculture. So mealworms are used currently as a feed for some exotic fishes. For aquaculture, I think if you use the mealworm from the biological recovery process, I think it's no issue. And also, I think PHA granules have been shown to have some prebiotic effect. I think there are papers published by some researchers and also patents filed to show that PHB can function as a prebiotic, which is good for the gut microbes. So there are, interesting effects to the animals. So feeding to animals, I think, should not be a problem. But to make it as a human food, I think, yeah, maybe there may be some, what do you call, resistance, or maybe some people don't like it. But I think, the process should try to use the mealworm to feed animals. Yeah. Then I think should be okay.
Thank you.
Yeah.
And I think it's time
Okay.
run very fast. So
Right.
thank you again,
Okay.
Dr. Saosuke Imamura, and also Professor Dr. Kumasude. That is very informative for us. And thank you again for sharing your time and your experience with us. And
Thank you.
yeah, thank you. And because we care about you, so I would like you to give us a bit time for survey. If you come to the virtual conference, you can see the survey, and then just please click on the post-seminar survey and give us some information to help the next time we can improve our conference to be better and better. And I think now it's time to take a break. And of course, in the afternoon section, we still have the oral presentation that related with the bioeconomy. So I would like to welcome everyone to join and the discussion and see the very interesting topic about bioeconomy in the afternoon section. And I would like to say thank you for everyone to sharing. I'm very appreciate and very happy to see our meeting today. And I think it's very fruitful and we can collaborate and think the more and more innovative for the bioeconomy in the futures. And so please have your lunch and we will come back again for the afternoon section. So we will start 12:45. So please enjoy your lunch and come back before the afternoon. So see you again on the afternoon section. Have a good day. สวัสดีค่ะ
Bye-bye.
Good afternoon, everyone. Welcome back to our section. That is the time for the oral presentation under the section of bioeconomy. And today we have 5 presenters that will give us the presentation. And I would like to telling some information about our presentation. All of presenters will have about 20 minutes for present, and after that we will have Q&A about 5 minutes. So first of all, that is the thank you again for coming on the ABB conference. And for our first presentation, I would like to ask Kun Thaksin Lersamran to sharing your slides, and then we can start our oral presentation. And during this time, I hope you enjoy your lunch. Be ready for our conference. So, Kun Thaksin, please sharing your slides.
Just a minute. You see my slide?
Okay. Okay. Again, you have 15 minutes for present and then 5 minutes for Q&A.
Okay.
And today, Kun Thaksin will give us the presentation under the title of the dynamic of non-structural carbohydrate and involved enzyme in relation with latex yield and rubber trees. So would you please give us some interesting information?
Yes, and everyone see my slides? You see
No,
my?
I could not.
Okay, just a minute. Yes.
You cannot see your slide. So,
You see my slide? Not yet.
not yet.
Um,
Shall we share it for you? If you could not.
okay, you can share for me. Sorry about
Okay.
for my computer.
Okay, so we will share the slide for you.
I cannot.
Wait a moment, please.
อืม
Hold on a second ค่ะ Wait a moment
พี่แชร์ไม่ได้คิดด้วยนะผมใช่ไหมเสร็จแล้วพอเลือกปั๊บมันขึ้นมาเป็นอย่างนี้
คือลงแชร์ก่อนแล้วเมื่อวาน
ไม่ยังพี่ยังไม่ได้ลองแชร์เลยไม่ลองลองดึงออกเลยคือไม่พิเศษ
and during we waiting for the sharing we have 5 oral presentation and of course for the presentation we will have the best award for oral presentation yes okay it's coming so it's your time คุณทักษิณ to present us you can see your slide now right
Yes, I can see my slide.
okay so please yes so it's time for you ค่ะ
Okay. 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 Thank you, Kun Thatka. So, may I introduce myself? My name is Thaksin Lersamlan. I am researcher from Kapi at Kasetsart University. Today, I would like to present you about my research topic, the dynamic of non-circular carbohydrate and involved enzyme in relation with latex yield of laboratory. Okay. It is well known that the laboratory is the economic plant of Thailand. It can produce the value product of latex. That is the income for the Thai farmer. Sucrose is the main carbon source for the latex synthesis. When we look inside the laboratory trunk, okay, you can see the sucrose from the leaf, yes, go into the phloem, and then it will send to the wood for storage starch. When the laboratory was tapped, storage starch will be hydrolyzed and send it through the vascular layer between wood and bark, and to go to the latex vessel in the bark for latex synthesis. Moreover, the dynamic of non-circular carbohydrate between wood and bark will be related with the enzyme activity. For the next slide, the group of enzyme activity such as sucrose hydrolysis and sucrose degradation using the enzyme of invertase and sucrose phosphate synthase we call SUSY. And then the next group is starch accumulation and starch hydrolysis. They use the AGPase and amylase. For the last group, it is for sucrose synthesis by using sucrose phosphate synthase, SPS. And then sucrose is the main source of carbonyl. If you go into the latex vessel for latex synthesis in the MEA pathway of the laboratory rubber particle. For the next slide. The question is, what are the dynamic of enzyme to control sucrose and starch metabolism and link to the latex yield? And then are there the difference between the tapping method in to the control with the activity of enzyme? To answer this question, we have to study the activity of enzyme and the content of sucrose and starch in bark and root between the difference of the tapping method. For the next slide, I separate for the parameter. We use the cone of RIM 600 is the laboratory cone, and then use the downward and upward tapping in the period of May and October. Yes, in untapped control, untapped control 3 and tap 3 on the panel of untapped panel and tap panel. And then we correct the sample. This is the bark and root. Next slide. This slide shows the sample correction. We separate into two parts of the enzyme assay and non-structural carbohydrate assay. For enzyme assay, we use the auger to collect bark. And then we put the increment boiler to correct the wood. The sample will keep it on the liquid nitrogen. And then they will extract and analyze the file of enzyme activity. For NSC assay, we use only increment boiler to correct bark and wood. The sample will keep it on liquid nitrogen also, and then they dye it by freeze dryer. Finally, the sample will extract and analyze the sucrose and starch content. For the next slide, we correct the latex, and we use the assay the content, the sucrose and inorganic phosphorus content in the latex by using the technique of latex diagnosis. And then we harvest the content of latex yield also. For the latex, we correct it and coagulate the latex by using the acid of TCA. And then we filter it and separate the serum. And we analyze sucrose and inorganic phosphorus by using the calorimetric assay. For the latex yield, is what measured by weighting the cumulative coagulate rubber. We call it cup lump in every month, and we propose it as the gram of latex per tree per tap per centimeter. Okay. For the result, the first result, we show the content of non-structural carbohydrate and enzyme activity between in bark and wood of laboratory, of the downward and upward tapping method. Okay, this bar chart shows the content of starch and soluble sugar, and this data we calculate the average of all parameters, such as the tree, untapped tree, panel, and the period of tapping cut, May and October. This bar chart show starch content which show higher than in wood than in bark. They show in the blue color. And then for the soluble sugar content, it show higher content in the bark than in wood in the both tapping method. And then for the next slide, for the enzyme activity, okay, we found that AGPS and amylase, it show higher activity in wood than in bark. On the board of tapping method, downward and upward tapping. Moreover, when we look for the enzyme of sucrose synthase and invertase, it shows higher than activity in bark, yes, than in wood. Contrary with the enzyme sucrose phosphate synthase, it shows higher than in wood than in bark. It indicated that enzyme AGPS amylase and sucrose phosphate or SPS is a main enzyme that locating in the wood and is related with the starch accumulation and starch hydrolysis and sucrose synthesis for the sucrose phosphate. And for sucrose synthase and invertase is the key enzyme that locate in the bark for the sucrose catalytic. And then it will use sucrose for the latex synthesis. For the next slide, I show you. Okay. This bar chart start in wood. It shows higher content in downward than upward tapping. Between, yes, in the downward and upward. I separate the parameter between downward and upward, and untapped T and tapped T, and untapped panel and tapped panel between May and October. This bar chart show that sucrose show higher content in the downward than upward. And then we will find that in downward tapping, it show higher starch content only in October on untapped panel of tap T. So this means that downward is induced for the starch accumulation only on the untapped panel of tap T in downward tapping. And then is linked with the enzyme activity of AGPase and amylase. I show, yes, below this bar chart. Okay. This part that AGPase show trend of increasing on untapped panel and tap panel on the tap T in downward tapping. The AGPase and amylase show in trend of increasing of enzyme activity from May to October. And then for upward tapping system, amylase show higher activity in October on both panel of the tap T. For the next slide, this bar chart it shows sucrose content in wood. They found that sucrose content show increasing trend in the tap T during high yield in the October of both tapping method. Yes, especially in the October, compare it between tap T and untapped T. We found that it shows higher activity on the tap T or in October. Yes, it's linked with the enzyme activity of sucrose synthase or SUSI. Is increased trend between untapped T and tap T on the both tapping method. Sucrose it show higher activity on upward tapping than downward tapping. However, but we did not find the activity of invertase, but we found a little bit of sucrose phosphate synthase in the wood tissue only in October of downward tapping. Okay, for the next slide, in the bark tissue, sucrose will show higher content in upward than downward tapping. And then we found that it links between, especially in sucrose synthase activity that increased from May to October on both panel of the tap T. And they found that sucrose synthase is a key enzyme on the both tapping method also. However, we found the chain of increasing of enzyme inverted on both panel of tap T only in the downward tapping system. And then for the next slide. Okay, for the next slide is the results of sucrose and inorganic phosphorus and latex yield. Yes, I analyze it in the latex. We found that upward tapping show higher latex yield more than downward tapping, especially in the October. And then it's linked with the sucrose content. We found that in upward tapping give the higher sucrose content than downward tapping. When we look inside the result of upward, we found that the sucrose it show decreasing content from May to October. It means that the laboratory that using for upward tapping can use the sucrose for catalyze and send to the precursor into the latex vessel for the latex regeneration. Then for the next slide. Okay, in conclusion, okay, we found that upward tapping show the higher to induce non-structural carbohydrate content and enzyme activity than downward tapping, especially in the sucrose content and enzyme sucrose synthase. This means that this tapping, upward tapping, give the higher latex yield potential, especially in the October more than downward tapping. And then sucrose synthase is the main enzyme that locating in the bark for the sucrose catalytic. It shows in the upward more than downward tapping. And then it shows, it means that sucrose will, the catalytic sucrose to support the raw material for the rubber biosynthesis. And for the ATPase, is play the role for start accumulation and amylase, which it show the hydrolyzed start in the wood. We found in the, actually we found on the board of tapping method. Okay, next slide. I would like to prefer a systematic pathway model of the enzyme mechanism and non-structural carbohydrate dynamic. For the first, sucrose from photosynthesis will translocate into the phloem, and then sucrose will go into the wood. First enzyme to catalytic sucrose is a SUSI. It will be produced. The main precursor is glucose 6-phosphate. Glucose 6-phosphate uses for the precursor for the storage start in the amylopase by using enzyme ADGPase. And then when the laboratory needs more to synthesize latex, the laboratory uses the enzyme amylase hydrolyze storage start in amylopase to produce the glucose, and then it's sent to sucrose synthesis by using enzyme sucrose phosphate synthase or SPS, and then sucrose will be transferred from wood to bark to the vascular layer. Yes. And then enzyme sucrose synthase and invertase will be analyzed, sucrose will be catalyzed and hydrolysis to produce glucose 6-phosphate. And then glucose 6-phosphate will use for the main source to produce the latex synthesis. Okay, finally, the last slide. I would like to thank Kapi of Kasetsat University and CLAT and the LVTNR to support the equipment and laboratory work. And then I would like to thank the Chachengsao Rubber Research Center, Rubber Authority of Thailand to support the sample and field work. Thank you for your kind attention.
Thank you, Ka. And if any question from the audience and committee, you can raise your hand or type on the chat box. And as I understanding your experiments related with the rubber quality, right? You need to improve your rubber quality by study the enzymes that involved for the rubber tree.
Yes, enzyme and
yes, could you emphasize which is the key enzyme that is really related with the latex yield and the properties?
Yes.
All 5 enzymes is important, or are there any specific or the key enzymes?
Sorry?
Are they important? I mean, you mentioned 5 enzymes.
Yes.
Are there all of 5 enzymes is important, or could you write a key enzyme that is very specific for latex properties?
Yes, I think this enzyme is really important and specific and linked to the latex yield production also. And I think this is key enzyme is sucrose synthase to catalyze the main source of sucrose. Yes, and then the laboratory will use the sucrose is a carbon source for the latex synthesis.
Okay, and one more question is about the season. I would like to know why you do the experiment on two season, right? It seems like
in May and October.
Yeah. How different between?
Yes. I would like to pick. I would like to compare between May and October, because in May is starting for tapping. It gives us for the, we call it is a low yield season. It gives us for the low quantity of latex, and then we correct in the October. It gives us for the high yield correction in the October. So that's why we want to know about the low yield and high yield, the effect of period season.
Yes. So.
Yes.
Yes. How about
Doctor? Yes, please.
Thank you. How about the sample that you, or your specimen that you take for analyze for the enzyme effect?
Again, please. The signal is not, I cannot hear you.
Okay. How many or specimen that you take for do the experiments? You mean that, for example, in May, so how many trees or how many something that you take that is the representative data for your analysis?
Okay. I use the three sample I use the 3 sample for each treatment.
You mean that you take one sample from one tree, or you mean that you take many trees from and accumulate for one sample? And when you analyze for the enzymes and for your this experimental designs, how you do I mean that how many samples you take? That means that for down tab and tab, so you take for one sample, but you take for how many trees from the only one crop, so you take this and another crops. Or you take in the same areas? How about your planning for the
Yes, I check the same area and I use total in each treatment I use the 3T for the untapped tea and 3T for the tapped tea. And when we correct for the season, we change the laboratory because the laboratory, yes, we change it because it is a lot of stem.
You take in the same area.
Yes, but I said it is the same area. But we change the laboratory. Yes.
Thank you.
Okay, thank you for the discussion. I think limit of time. So even the interest information, maybe we can contact each other later. And thank you for Kun Thaksin for your presentation. And I would like to move to our oral presentation. The oral presentation too, that is from Kun Atit Pongtip, under the title of the physiological response and sugar partitioning mechanism of mice at flowering stage under different drought stress regime. And please share your screen. And Kun Atit come from the Department of Agronomy, Faculty of Agriculture, Kasetsart University, Thailand. So now I give your time for presentation, please.
Thank you, Chairman, for your introduction. Good afternoon, ladies and gentlemen. Today, I would like to present on the topic of physiological response and sugar partitioning mechanism of maize at flowering state under different down state regime. Recently, the demand of maize in Thailand has increased, but maize production of Thailand cannot increase due to the limit of the planted area. So the research of increasing the plant area at late rainy season and dry season is important and interesting nowadays. Downstate is important problem from maize productivity at late rainy season and dry season. The primary tissue with downstate was the main effect when, how serious, and how long. And today I will concentrate on serious of downstate. Currently, at all study effect of irrigation level show that downstate at 150 mm evaporation from Class A plant had decreased total yield component such as number of leaf per year, number of seed per row, and 100 seed weight. And Patasali et al. Show that photosynthetic rate of maize at all development stage of maize were decreased under down state at 50% and of water requirement of maize production. So the objective of this study was to evaluate effect of different downstate regime and varieties on physiological response, sugar partitioning, and gain yield production of maize. The experiment design was split pot in RCBD. Main pot was 4 different downstate regime. Subpot was 2 varieties of maize with 4 replication. In the main pot, there were 4 different downstate regimes, which were 20, 30, 40, and 60% soil moisture content. In the subpot, there were 2 varieties, which were Nakhon Sawan T and Suwan 4452. Figure 4. The experiment procedure under maize developmental state. After critical tassel initiation state was controlled at 60% of soil moisture content. At critical tassel initiation state was start of downstate condition. Downstate condition was controlled at 40, 30, and 20% of soil moisture content for 7 days on downstate treatment. After 7 days of downstate condition, will begin to the data collection and after that all treatment were controlled at 60% of soil moisture content. Data collection, crop growth rate, leaf area index, kilo per year, number of gain per kilo, 100 seed weight, gain yield were correct at harvesting time, and leaf relative water content, net photosynthetic rate, transpiration rate, stomata conductance, total soluble sugar content of leaf, stem, and seeds were correct at 7 days after downstate. Then analysis of variance was carried out using the static software package version 8 and the mean comparison was done by LSD method. The experiment was conducted at in-house Department of Bioeconomy, Faculty of Agriculture, ASEAN University, Bangkok, Thailand, from November 2018 to March 2019. The mean of minimum temperature was 24.9 degrees Celsius. Mean of maximum temperature was 33.3 degrees Celsius, and mean of average temperature was 29.1 degrees Celsius. Next, I would like to present result and discussion. Table 1 show analysis of variance of leaf area index, crop growth rate, gain row per year, number of gain per row, 100 gain weight, and gain yield. There are significant difference in all parameters among different downstate regimes. And for variety factor, there are significant difference in gain row per year. Table 2 show analysis of variance of leaf relative water content, photosynthetic rate, transpiration rate, stomata conductance, total soluble sugar content of leaf, stem, and seeds. There was significant difference in all parameters among different downstate regimes. For effect of variety, there were significant difference in leaf relative water content, total soluble sugar content of stem and seeds, and there was significant interaction effect of downstate regime and variety on total soluble sugar content of seed. Figure 6, effect of different downstate regime on crop growth rate and leaf area index show that 20% of soil moisture content show low rates of crop growth rate and leaf area index. Figure 7. Effect of different downstate regime on photosynthetic rate, transpiration rate, and stomata conductance. Show that 20% of soil moisture content had lower photosynthetic rate, transpiration rate, and stomata conductance than those in 60, 40, and 30% of soil moisture content. Effect of different downstate regime on leaf area index, crop growth rate, and cane yield, which were decreased at 20% of soil moisture content due to the decrease of photosynthetic rate. Hudgen et al. Report that decrease of photosynthetic is controlled by non-stomatal mechanism of gas exchange connect with damage of mesophyll cell, membrane, and chloroplast, and visible in assimilate, synthesis, and transport. And Musa report that decrease of photosynthetic rate cause of stomata causes reduce carbon dioxide oxygen ratio in leaf, inhibits photosynthetic, and stomata conductance decrease. Figures 8. Effect of different downstate regime on leaf, relative water content, total soluble sugar content of leaf, stem, and seal show that 30 and 20% of soil moisture content had high total soluble sugar content of leaf, total soluble sugar content of stem, more than 60 and 40% of soil moisture content, and 30 and 20% of soil moisture content had low leaf relative water content, total soluble sugar content of silt, more than 60 and 40% of soil moisture content. Downstate reduce photosynthetic rate, stomata conductance, and transpiration rate by 20% of soil moisture content. The relative water content of leaf influence storage of soluble sugar content at the leaves and stem, but it also influence transfer of soluble sugar content of silt. Serlat and Cynthia report that sugar partitioning mechanism at downstate, accumulation of organic and inorganic solute of stem and leaf, maintain low water potential, decreasing actual water content, and mechanism was osmotic adjustment or osmoregulation. Figure 9. Effect of different downstate regime on yield component show that 20% of soil moisture content have low gain low per year, number of gain per low, 100 gain weight, and gain yield, more than 60, 40, and 30% of soil moisture content treatment. Since Selmayer et al. Report that sufficient sugar supply in required for silt and ear development, which lead to the potential of seed setting. And this et al. Report that and silt execution begin early and the pollen tube is unable to reach the ovary. The resulting is no fertilization. Figure 10. The correlation of gain yield and total soluble sugar content of stem and seal show that gain yield was decreased by high of total soluble sugar content of stem, but gain yield was increased by the high total soluble sugar content of seal. And for variety, table T effect of main variety, maize variety on kilo per year, leaf relative water content, total soluble sugar content of stem and seal show that Suwan 4452 variety significant increase kilo per year and total soluble sugar content of stem. So, conclusion. Downstate legume at 40 and 30% of soil moisture content, maintain physiological response, maintain sugar partitioning, can maintain gain yield. But downstate legume at 20% soil moisture content provide low leaf aerobic, cocoa rate, photosynthetic rate, total soluble sugar content of silt and yield component. And Suwan 4452 was the optimized as made variety for downstate tolerance. That I would like to conclude the main of soil water level maybe it can to improve late rainy season and dry season made productivity in order to increase made gain supply for bioeconomy expansion based on made demand. Finally, I would like to thank Educate Study Development Scholarship from the National Research Council of Thailand for Research Fund, Faculty of Agriculture Kasset University for Experimental Field, and Plant Physiology and Renewable Energy Laboratory, Department of Agronomy, Faculty of Agriculture Kasset University. Thank you for your attention. Thank you.
Thank you for your nice presentation. That's all about the maize. That is very pricey. And then it seems you try to develop how drought rate affect for the maize productivity. But may I start some quick questions? Actually, I'm not in the field, but I'm quite interesting. I'm curious to know about how we can optimize the maize production. Especially, it seems we need the water not less than 20% to maintain stable production. That is correct from my understanding. Do you have any idea or give me some more suggestions?
Some water. Excuse me, I would like to show some slide.
Yes.
Today have a technology for control soil moisture content and use machine for control soil moisture content in field, and you can manage for water in the soil for low moisture content.
So it's been real time
monitoring. Real time monitoring.
Wow.
Yes.
Okay.
Yes,
so
smart farming. Yes.
Okay, it's good to know. Yeah, for the very advanced technology that can monitoring on site, and we can manage our production crop.
Yes.
It sounds very good.
And other technology
any...
use evapotranspiration ET for calculate for transpiration of water or transpiration of pen and calculate and water on the soil. Okay.
Yes. Okay.
Nice
Are there any
question.
questions? Are there any questions from our committee or any presenter? That's welcome. And during waiting for the next question, is it okay? We have the question that if this hypothesis can be applied for sweet corn or other species.
For sweet corn on other species of corn different for require of water management. And for this study, can use only for maize because for sweet corn and baby corn, them need and this need more water for maize.
But maybe it can use some technique related, but it has some specific for each species of corn, right?
Yes.
Yes, but I think it's a good challenge to learn and to observe what is the key factor, what is the key parameter that we can monitor for a very good production. And do you have any idea during the flooded? Now we face for the flooded. Are there any criteria when you would like to produce the corn under the flooded or rainy season? Do you have any idea?
I have idea. For this figure show that total soluble sugar content of cereal control to gain yield. Maybe you can control total soluble sugar content of cereal. You can promote to high gain yield in production.
Thank you for your suggestion, and I think it's a limit of the time for discussion. And again, thank you, Kun Atit, to give me a very nice presentation. And the next presentation, I would like to introduce Kun Nichakon. She will give us the presentation under the topic of genetic diversity and physiological characteristics of quinoa under different ecotype conditions. So please, Kun Nichakon, to give us, and you can share your slide.
Thank you, Chairman. And good afternoon, everybody.
Yes, now your slide. Okay, we can see your slide clearly.
Okay. I am Nichakon Khatkao. I would like to present genetic diversity and physiological characteristics of quinoa under different ecotypes. At first, I would like to introduce quinoa. Quinoa was originated in South America. Cultivation has expanded in many continents, such as America, Europe, Asia. Because quinoa can grow and produce good yield in dry and cool weather, they want moderate rainfall and relative humidity. Moreover, quinoa have many benefits for your health, such as high protein content, high fiber, good for heart, and weight control. And quinoa was added in superfood too. Moreover, quinoa was tolerant to extreme soil and unsuitable conditions. The justification of this title was from the problem of highland agricultural system. In the northern part of Thailand, most of agricultural area were cultivated under rain-fed conditions that affect limiting water. So farmers want to expand agricultural area for cultivate much more because the farmer can cultivate only one time per year. That's why in the northern part of Thailand can find out the intensive slash and burn and shifting agricultural system, cause of poor soil fertility and deforestation problem. How to reduce this problem? We want to make the alternative plant for farmer. That plant can cultivate in late raining time and they have costly price in market. Thus, the understanding of interaction between genotype, covariables, and environment and phenotype adaptation was very importantly required. According to Tatara 2012 reported, the mean seed yields of 5 quinolines, these 5 quinolines, was grown at 4 locations in Central Asia. This diagram shows the different physiological response in form of seed yield in each location. These results explained why the studying of different ecotypes was necessary. Previous research, Pitipong 2017 reported suitable location for planting quinoa in Thailand. Each area was different by sea level, soil texture, and average temperature, especially Pangda and Prabat Hoi Tong. There were 700 and 400 above sea level. There were loamy soil and sandy soil. Moreover, suitable season was late rainy and late winter. According to the phenotype adaptation, we need to group the genetic diversity of quinoa accessions by using unweighted pair group method with arithmetic mean. Tatara 2012 reported dendrogram showing 35 quinoa accessions in the UPGMA cluster analysis. Population were represented in 4 major clusters. Therefore, the cluster analysis could explain the genetic correlation which relate to the final ecotype response. The objectives were to study genetic diversity of quinoa accessions and to evaluate phenotype yield of quinoa under different ecotypes. The experimental design was randomized complete block design, combined location with 4 replications. Treatment was quinoa 24 accessions. There are 3 locations. We use method is AFLP method and GGE bipod. These were the picture of quinoa 24 accessions. These three locations were different by sea level, mean, maximum, and minimum temperature, relative humidity, average rainfall, soil texture. In field, we collect physiological traits. And next one is harvesting trait. And after that, in the laboratory, we use AFLP technique for study the polymorphic of DNA in each accessions. There were 5 periods for data collection. The physiological parameters were used for cluster analysis to group the 24 accessions which followed by their phenotypic response. Locations are Ban Hoi Tong Royal Project at Lampung Province, Royal Agricultural Station Pang Da, and Doi Pui Research Station Kasetsat University at Chiang Mai Province. This research had duration approximately 2 years. This table showed the mean seed yield of 24 quinoa that was grown in 3 locations. Look at the accession number 18. KU18 had a table of mean seed yield in 3 locations. The table 2 shows the result of correlation analysis of physiological traits. At the state of harvesting period, the growth parameter showed the positive correlation such as yield, crop growth rate, dry weight, and harvest index, while some parameters showed the negative correlation response, such as net assimilation rate, leaf area index, and leaf area ratio. Total dye weight and crop growth rate had a strong positive correlation to yield, as same as harvesting index. This suggestion describes the major factors to promote yield productivity were accumulation of dry weight and high crop growth rate, while the leaf area ratio was the secondary parameter which negative correlated to harvesting index. Generally, the fall of senescent leaf during harvesting period could make the greater ratio of grain. Therefore, hierarchical cluster analysis arranged the quinoa 24 accessions into 3 groups. The comparison of UPGMA between DNA and interaction of DNA and physiological traits showed the different pattern of cluster caused by environment effect, which explains the different ecotype of 24 quinoa accessions in each location. The stability analysis using GGE by plot method to assess the stability of quinoa yield characteristics. The suitable location for planting quinoa was Doi Pui and KU23, this one. And KU23 had good yield potential at Doi Pui. Moreover, KU18 was suitable for planting at Hoi Tom and Pangda. And KU6, KU11, and KU21 show the greatest potential to adaptability in all locations. The comparison of UPGMA between DNA and interaction of DNA and physiological traits showed different pattern of cluster caused by environmental effect, which explains the different ecotype of 24 quinoa accession in each location. Conclusion: UPGMA cluster analysis by DNA could identify the 24 accessions of quinoa into 3 groups, and KU18 was the highest potential to physiological adaptation and yield production in Hoi Tom and Pangda. Why? KU23 was the most suitable for plantation in Doi Pui. KU6, KU11, and KU21 were the most adaptively in all locations. The further work, the breeding program, which target to enforce the quinoa production in geometrical profile, like Hoi Tom and Pangda, could be used. The genetic base which closely related to KU18. I would like to thank you, National Research Council of Thailand, Royal Project Foundation, the National Genetic Resource Republic of Chile, the Embassy of the Republic of Chile, Princess Sirinthorn International Center for Research, Development and Technology, Ban Hoi Tom Royal Project at Lamphun Province, Royal Agricultural Station Pangda, Doi Pui Research Station, Kasetsat University at Chiang Mai Province, Department of Agronomy, Faculty of Agriculture, Kasetsat University, Faculty of Agriculture, Kasetsat University, Faculty of Agriculture, Department of Crop Science, Gerd August University of Göttingen, Germany. Thank you for your attention.
Thank you for your nice presentation. And we know a lot about quinoa, yes. And it seems we can plant quinoa on the highland of Thailand now. And it seems a lot of variety of quinoa, KU that you try to breeding and try to develop and taste on the north part of Thailand. And it seems like your collaboration with a lot of team, like the farmer who in the many highland in Thailand to planting your crop. So how long for one crop when you do the experiment?
We plant we use 3 years or 4 years to plant per one crop. Yes, but we the proper climate, the proper season to plant is late rainy and winter. Around this is around the October, November we can start to grow, and we will harvest it on maybe January. The weather does not have rainy, because it will make the seed, the humidity will destroy seed, and we cannot control the เชื้อรา the fungi yes
Yes. Mhm. And do you find, are there any genetic diversity that you try to mention? What is the genetic diversity of the KU19 that make it better than another? Do you have?
KU18
Yes, you make the conclusion that KU18 have a very high yield production in all locations. What is the genetic parameter, or what is the key function why KU18 is the best? Do you have any evidence?
we will
Or you will do for the next experiment to get more knowledge
yes,
about this
now we
one?
just see it only we will determine on the seed yield of 3 location and we will เขาเรียกอะไร we เข้าโปรแกรม the GGE Bipod and that show the stability of quinoa accession that the KU18 was the best. It's not the best in every location, but it have a high yield in 3 locations.
Mm-hmm. And have you ever tasted your quinoa after you planting? It is tasty.
It's like
Yeah.
eating.
no,
Yes.
yes.
If we can eat it with rice, with everything that we eat, because it don't have a taste, it don't have a strong taste. It seems like เขาเรียกอะไรถั่วพวกบีน or something like เม็ดแมงลักอะไรแบบนี้ค่ะคือมันจะไม่มีรสชาติอะไรเลยแต่ว่ามันจะแบบสามารถกินได้กับทุกสิ่งสามารถกินกับขนมก็ได้สามารถกินกับอาหารก็ได้ Can I answer in Thai? Thank you.
And it seems not much tasty, but it's very good for healthy, right?
Yes, it's healthy. It have a lot of fiber. So if you eat in proper amount, it just only 1 spoon or 2 spoons to mix with another food that will okay with your health per day. Yes.
Okay. I think nowadays we familiar with quinoa we can find from the market, and I think it will be an alternative for the people who and who be a healthy people who don't want to consume a lot of starch, but they try to looking for an alternative. And quinoa, I think it's one trendy cereal that will be come into the future, and I think it's a good research also that we try to develop and move our quinoa to planting in our ASEAN country. And because individual locations, they also have the some limitation. But finally, you can looking for one promising crop that maybe in the future we can see KU18 launch on the market. So how you can keep going. And thank you everyone again. And I would like to move to the next presentation. That is also from Kun Panapat Prompen. And Kun Panapat Prompen will give us the presentation about utilization of banana peel biomodern for dyeing silk fabric with marigold flower extraction and its UV protection property. The title is very interesting. So I would like to give this time for Kun Panapat to give us the more detail about this topic. Yes, please.
Yes, thank you, Chairman, and good afternoon, everyone here. I am Panapat Prompen from Faculty of Agroindustry, Kasetsart University. I'm glad to meet all of you here today. And today I represent our research project, which was collaborated with Assistant Professor Sirinan Kentong from Faculty of Science, Srinakarin Road University. Today, I'm going to present about the utilization of banana peel mordant for dyeing silk fabric with marigold flower extraction and its UV protection properties. In this presentation is divided into 4 parts, including introduction, materials and method, result and discussion, and finally conclusion. Let me start with the introduction. In Thailand, cultivated banana or Kloi Namwa is widely planted in Thailand. And because of banana contain high nutrition, so it can be processed to various types of food. And after that, there were loads of banana peel waste after food processing. And in the past, there was some previous study about the banana peel extraction, and it was found that banana peel is a resource of phenolic compound. And in the process of dyeing textile with natural dye, tannin is playing an important role because of tannin contain phenolic compound, which can be used as a mordant in natural textile dyeing process. So that we found that banana peels contain tannin, and we try to use the banana peel as a dye or mordant for dyeing textile material. And it also found that the resulted fabrics from dyeing or mordanting with tannin, the fabric can be resistant to UV radiation and also resist to bacteria such as E. coli, S. aureus, or K. pneumonia. Moreover, the marigold flower has a color of yellow to orange. Is mostly used for decorate places or used in the worship in the Buddhist or Hindu temple. And at the end of the day, there will be loads of marigold flower waste as well. And previous research show that marigold flower contain carotenoid and flavonoid. So this kind of chemical can be used as a dye for dyeing textile as well. And there was some previous study show that using the marigold flower as a dye for dyeing textile material with the chemical mordant, it showed that the color strength and color fastness of the dye fabric were improved. So in the purpose of this study, we try to optimize the mordanting parameter, including time and temperature for dye-using fabric by using banana peel extraction as a biomordant and using marigold flower as the natural dye. And after that, we try to evaluate the color fastness and the UV protection property of the dye fabric. Next one is the material and method. Let's start with the banana peel preparation. The banana peel preparation was carried out by chopped fresh banana peel into small pieces like 1 times 1 centimeters, and then bring them to boil in water at 90 degrees Celsius for 60 minutes by keeping liquor ratio at 1:3. After that, the mixture was cooled down and filtered, so we can get the banana peel extraction for the further study. Later, we try to prepare the marigold flower dye extraction. We separate the marigold flower dyes from the petal from the flower, and then bring them to boil at 70 degrees for 30 minutes by keeping liquor ratio at 1:20 according to the previous research. After that, the mixture was cooled down and filtered, and we got the marigold flower dye extraction. For the dyeing procedure, we have 2 dyeing procedures. The first one we call is the pre-mordant method. Me describe this procedure. To start with, the silk fabric were immersed with the banana peel extraction at liquor ratio at 1:50. At this time, we differentiate the time and temperature at 30, 60, and 90 degrees Celsius with 60, 90, and 120 minutes. After that, the mordanting fabric will be exhausted in the marigold flower extraction at 90 degrees for 60 minutes by keeping liquor ratio at 1:50 as well. After that, the dye fabric were rinsed by the running water for 10 minutes and dry at room temperature for 24 hours. The second dyeing procedure is called post-mordanting. In this procedure, begin with dyeing silk fabric with marigold flower extraction as liquor ratio 1:50 at 90 degrees for 60 minutes. After that, the mordant were applied by the different time and temperature at 30, 60, and 90 degrees Celsius by the time at 60, 90, and 120 minutes. After that, the dye fabric were rinsed by room temperature by running water for 10 minutes and dry at room temperature for 24 hours. Later on, the testing method, according to the KOS value that we use as a tool to decide which condition is the optimal condition, so that we measure the color of the dye fabric by using the Hunter Lab spectrophotometer, and the fastness of the fabric was tested into different kinds of fastness, including watt fastness and light fastness. And later, the UV protection property were evaluated by the AATCC 183 2014, so that we can get the UV protection factor or UPF. Result and discussion. As can be seen in table 1 that the CT is the control fabric, which is no mordant at all. And we can see here that at 30 degree or room temperature, the KOS value were lower than the control one. So this means the room temperature was not suitable for dyeing silk fabric. And we can see the trend here, like if the temperature increase, the KOS increase, while the L value, which is represent the lightness of the fabric, decrease when the temperature increase. Moreover, the B value, which show the yellow and blue of the fabric is also decreased when the temperature decrease. So this table is about the pre-mordanting method. Let me move to the second table, which is the post-mordanting method. It can be seen here that at 90 degrees Celsius and the mordant time at 120 minutes, the KOS is the highest one. So that means in this temperature or the higher temperature and higher duration time, when put the fabric into the mordanting bath, it will enhance the dye uptake to the silk fabric. So we can conclude from this table that the optimal condition for dyeing silk was at 90 degrees Celsius and the duration is 120 minutes. So the color fastness of the dye fabric, after we choose the best condition or optimal condition from the previous step. So we use 9 PO, or this means the post-mordanting and dye mordanting at 90 degrees for 120 minutes. We can see that the wash fastness of the fabric did not improve from the no mordant fabric. However, the light fastness also did not improve as well. So let's move to the UV protection property. In this UV protection property, we choose the 3 samples. The first one is undyed silk fabric. The second one is the dyed silk fabric with marigold dye but no mordant. And the third one is the dye with silk dye fabric with marigold flower and post-mordanting at 90 degrees 20 minutes. And in this slide, the UPF value can be used to classification the UV protection property. And if the UPS value lower than 15, it classified as poor protection. So it can be seen here that the undyed silk fabric show the poor protection both in dry condition and wet condition. However, the dyed silk fabric and the dye and modal silk fabric show the higher value. And in this case, if the UPF value was higher than 40, is classified as excellent UV protection. So from this table, it can be concluded that using the banana peel as a biomordant, dyeing the silk fabric can make the fabric resist to UV radiation. So the conclusion from this research, we can conclude that we can use banana peel waste as the biomordant for dyeing silk fabric with marigold flower dye, and the optimal condition was achieved at 90 degrees Celsius for 120 minutes by post-mordanting method. However, the wash fastness and the light fastness did not improve here. And in this case, it might need to use the fabric preparation before the mordanting method to improve the wash fastness and light fastness. And the last one, we can conclude that dyeing silk fabric with banana peel biomordants with marigold flower dye can improve the UV protection property of silk fabric. And thank you. We are gratefully thanks to the Department of Home Economics, Faculty of Science, Srinakarin Virod University for providing the facility used for this study. And this is the list of reference. And thank you for your attention. Thank you.
Thank you for your presentation. That's very interesting topic. And then it's one that we can find banana peel also have a high value. Even we just leave it and get it like the trust, but however, it's a value if you know the best technology to extract and useful something for like a dyeing, and interest to know that it also has the UV protection also. So
Yes.
yeah, it will be very interesting. So are there any questions and comments from the floor? Now it's welcome everyone to share and talking with our presenter. It's very interesting to know. And now you still more development for the dyeing.
Yes. Yes.
Are there any material from
I
food waste?
I didn't do the improvement in the food waste, but I also make more tests like antibacterial to evaluate that is there any properties like antibacterial.
Okay. So please, Dr. Rangsimai, to give some question and comment.
Thank you, Dr. Panapat, about your presentation.
Yes.
Hear me?
Thank you.
Okay. So, how about the concentration of tannin that you extract from the banana peel? Did you measure it?
Yeah, actually, the tannin contents.
Yeah.
The tannin concentration.
Yeah, because
Yes.
you said that you extract, but you still be in like a liquid, and you prepare for like a post modern and pre modern, but you didn't do like a dry dye weight or dye materials of tanning. So I
Yes.
would like to know the concentration, because if not, you cannot control it when you do for the next experiment.
Yes. Actually, I have the previous work about the tannin extraction and evaluated the tannin content in the different condition that extract from the banana peel. Like I differentiate the time and the temperature of extraction, and it was found that in this condition, in this condition, at 90 degrees for 60 minutes was the optimal condition for extract the banana peels.
Yeah.
Yes. So yes, sorry that I didn't cite myself in this presentation.
Okay. So it means that when you use for the next time, you can control the concentration that you use for the post modern.
Yes.
At your conclusions.
Yes.
You compare with the commercial tanning?
Not yet. That's a good idea. Thank you very much. Yes.
Thank you. And we have some questions from the audience also. That have you compare with the other commercial modern? Do you get the questions when you compare? Yeah.
Not yet. Yes, that's an interesting topic that I will do in the future. Thank you very much.
Okay. Yes. So if you develop something new, it's very useful. So maybe we have to benchmark with the something commercialized. I think, yeah, it's very interesting too.
Yes. Thank you.
Okay. Yes, and any question and comments we still have time for all of the comments and questions. If you would like to share or comments, you can raise up your hand or can put the message on the chat also. And I have a quick question.
because it's interest to develop the nature dye. It means extract
Yes.
from the nature, but how to
Yes.
control the dye color? Is it some limitation, or are there any rule for control the color?
Yes, that's the problem when we try to repeat the experiment, because if we get the material or the waste from the different place or different season, the yield or the color might be different. Yes, so I think the best way to prepare the color, it might be extract the flavonoid or carotenoid from marigold flower and dry it or make it as a powder and keep it in as the powder substance, so that might can control the yield and the color of the substrate.
Yes, and are there any application in our tree or any location for use banana and marigold?
Yes, actually, this banana, I got this banana from Nakhon Nayok province, because in Nakhon Nayok there are lots of banana, and for the marigold flower dye in this study, I bought it from the market, and maybe in the future I might go to collect the flower from the temples, so that can be clear the waste from the temple.
Yeah, I think so, because a lot of the marigold from the temple. Yes.
Yeah,
it's a good idea to make it more and more value.
Yes. Okay. Thank you.
Let's see, are there any questions and comments from our audience? Yes, we run on time. So thank you, Dr. Panapat. And the last presenter for today, I would like to introduce Kun Sirinthorn Kaewchoi, the last presenter for our section today. She will give the presentation on the topic of effect of source sink balance management of an optimum harvesting of maize seed, vigor and storability. So please sharing your slide, and then you can give us your presentation. Okay, yeah, clearly, we can see clearly, so please make the slideshow. Yes.
Thank you, Chairman, for your introduction. Good afternoon, ladies and gentlemen. Today, I would like to present in topic of effect of sourcing balance management and optimum harvesting time on seed vigor and storability. Introduction. Sourcing relationship on your productivity. Borat and Otekul reported that variation in final grain weight reflected in interaction between source capacity and sink strength during the effective grain filling period. Gifford et al. Reported that middle leaf of the stem had a greater important role than the outer leaf because of the greater surface for light absorbing in the photosynthesis. Complete defoliation was led to minimum seed yield because of decrease in seed weight and field grain percent. When Denberg et al. Reported that distance of leaf and ear width participant in photosynthetic efficiency were important in slide defoliation. Top leaf of the ear transfer about 23 to 91% of photosynthesis to the crop, and the greater number of transfer material belong to the least leaf or short period. of maize leaf defoliation up to 50% did not have an adverse effect on maize grain and yield components. Effect of harvesting on seed quality. Mehta et al. Reported that seed development was the period between fertilization, maximum phase weight accumulation, and seed maturation. It began at the end of seed development and continued till harvested. Seed yield and quality also depending on the proper harvesting time. Figure 1 shows schematic of seed development and seed quality change. Bewley and Black reported that harvesting time of any crop for seed quality was depends on their maturity time and physiological maturity. Optimum harvesting periods help to obtain better quality seeds. Harvesting state influences the quality of seed, which relate to germination, vigor, viability, and also strawability. Junior et al. Reported that a delay in the harvest also increased the potential demand for all excessive rain during seed post-harvest processes. Toon and further reported that strawability of seeds was influenced by pre-straw history of the seeds, seed maturation, and environmental factors during pre-harvest and post-harvest. For all reasons I have said, this research of the objective of this study was to study the effect of sourcing balance management and harvesting time on seed vigor and storability of maize. Material and methods. The experiment was arranged into speed plot in randomized complete block design with 4 applications. The experiment treatment were main plot, which was the defoliation pattern with 5 levels. Were D1 equal control no leaf removal. D2 equal defoliating of leaf. D3 equal defoliating leaf below the ear. D4 equal remain to top leaf. D5 equal remain subtending leaf of the maize. Leaf cutting days with 13 days after silking. Subplot had 2 different harvesting time were H1 equal 100 days after emergence and H2 110 days after emergence. After each harvest, samples containing 10 ears were placed in paper bag and then taken to a hot air oven for drying at 40 degrees Celsius. The drying was performed until the seeds reaches approximately 12% of moisture content. Seeds were stored in plastic sealed bag in 25 degrees Celsius. Then seeds were sampling for seed component, seed quality, and vigor was tested at 6 months. Data collection. The experimental data were recorded with following yield components and yield parameter and seed quality and seed vigor parameter. Plate and duration. Former field at Utong, Suphanburi Province, Thailand, and then I did my laboratory work as crop physiology and renewable energy crop laboratory, seed laboratory, and Bioeconomy Department, Faculty of Agriculture, State University. Duration December 2018 to July 2020. Result and discussion. Table 1 show analysis of variance for ear weight, row number per ear, seed number per row, seed number per ear, and 100 seed weight. From the table 1, you will see all parameters were significantly different under the effect of defoliation pattern and harvesting time. They are significantly different in ear weight. Complete defoliation severely reduced ear weight. Defoliating leaf under of the ear has greater ear weight than removing top leaf of the ear. Harvesting time in 110 days after emergence. Show the highest ear leaf. Complete defoliation severely decreased row number per year, seed number per row, and seed number per year. Reduction of supply assimilation by defoliation and significantly reduced row number per year, seed number per row, and seed number per year. 100 seed weight, the results show removing all leaf severely reduced seed yield. D1 and D3 were the best increase high 100 seed weight. Table 2 show interaction between defoliation pattern and harvesting time. This significantly altered row number per year. Interaction of D1 H2 and D3 H2 show the highest row number per year, which was statistically significant. Table 3 show analysis of variance for seed germination. For seed germination percentage, shot length, root length, seedling growth rate, speed of germination, and ASH germination at 6 months. From the table 3, you will see all of parameters. They are significantly different under the effect of defoliation pattern in seed germination percentage, shoot length, seedling growth rate, speed of germination, and ASH germination at 6 months. Harvesting time, they are significantly different in seed germination percentage, speed of germination, and ASH germination at 6 months. D3 give the highest seed germination percentage. It might be due to that defoliation as an environmental stress can reduce seed germination percentage and described by Hyderich. According to harvesting time in 110 days after emergence show the highest seed germination percentage. Matthew reported that the lesser germination percent in early harvested seed may be due to presence of more number of immature and unfilled seed. Defoliation treatment had a significantly impact on seedling shoot length. Hyderich reported that they had a negative correlation between root length reduced while shoot length, seedling growth rate, and germination percentage increased. Seed vigor, the significantly effect of defoliation treatment on seedling weight and vigor was shown. D3 the best increase high seedling growth rate, speed of germination, and ASH germination percentage at 6 months. Of storage is not significantly different from D1, D4, and D5. Discussion. Defoliation leaf under ear D3 severely increased ear weight, seed number per row, seed number per ear, shoot length, seedling growth rate, speed of germination, and seed vigor. Hyderich reported that this might be due to those defoliating leaf under ear acting as a parasitic and metabolic sink that's competent for ear and kernel development during the grain filling period. Those leaves as below parts of the maize stem and upper leaf whose provide shade for the leaf in the central position. Defoliation leaf on top of the ear D4 and D5 cause more impact with decrease in the rate of grain filling because of only remain leaf were unavailable to supply enough to requirement of assimilate for plant. The effective period of grain filling has greater affected increasingly by defoliation than the rate of grain filling. Hyderich reported that the results suggest that the top leaf could be prevent to defoliation because this treatment show the negative effect on yield. Harvesting state of maize seed at 110 days after emergence markedly increased speed of germination and ASH germination percentage. After at 6 months, Gone SO reported that increased speed of germination and ASH germination percentage might be due to maturation of seed in the state, resulting the improvement of germination percentage and seedling length. Conclusion. Therefore, improving the seed quality in maize should focus to promote the post-flowering sourcing ratio. Leaf development, leaf defoliation of upper of the ear was more impact to all investigate characteristics as well as the result that suggests the upper leaf should not defoliate because this treatment had negative effect on the yield. To sum up, it can be concluded that harvesting of maize seed at 110 days after emergence would be physiologically mature seed that and ultimately led to the highest seed quality as well. Quality seed production at the appropriate time and seed maturity are a must for successful crop production. Acknowledgement. Thank you to manager and staff of Syngenta Seed Thailand Corporation, former field at Utong District, Suphanburi Province, National Research Council of Thailand, Economy Department, Faculty of Agriculture, Kasetsart University, and Professor Dr. Michael Bierwehr, Georg University of Göttingen, Germany. Thank you for your attention.
Thank you for your presentation.
Okay, that also time for us again to make a question to the presenter. And today, of course, we know a lot of the maize after we harvesting and until how we store it. So I think we engage the knowledge about this opportunity for our crop also. So let's see, are there any questions from the floor, from the audience? And in general, how long, in general, when we do storage the maize before we make some another process? Hello. ค่ะ Do you hear?
Again, please.
In general, when you harvest, and then how long for storage in general for the maize before you launch or sell it? How many days for storage in general?
6 months.
6 months? Yes, and sometimes we heard about the long-time storage. Maybe it has some contamination also if the storage is not good, like some alpha toxin when the fungal coming to the storage tank. So about this effect, do you have any idea to protect the toxicities of the maize?
The drying of perform until the seed reaches approximately 12% of moisture contents. Seed was stored in plastic seal bag in 25 temperature.
Yeah, and it means your set can keep the and storage at least 6 months, right?
Temperature relative humidity,
Okay. Are there any questions and comments? It's welcome. I think you do a lot of experiment to gain the information of the maize production and storage. And what is the future plan for the maize seed storability? Do you have any future plan to make it more sustainable or more effective? Do you have any idea? To improve your technique or make more sustainable technique? Do you hear me?
มันก็หนูขอตอบภาษาไทยนะคะ
ใช่ค่ะ
ค่ะคือมันก็จะเก็บเกี่ยวได้เร็วขึ้นนะคะคือถ้าสมมติว่าเราต้องการคุณภาพใช่ไหมคะแล้วก็ถ้าสมมติว่าบวกกับการที่เราจัดการซอสที่ดีอะค่ะจำนวนใบที่ดีมันก็จะมีผลต่อการส่งอาหารไปที่เมล็ดค่ะแล้วก็บวกกับการเก็บเกี่ยวที่ดีเนี่ยมันก็จะมีผลต่อการเพิ่มคุณภาพแล้วก็ปริมาณของเมล็ดด้วยค่ะ
ค่ะ thank you ค่ะ it means every step gonna be very effective and you need to care every step, right? Since from your planting to make it consume a lot of nutrients and finally you will harvest a very perfect crop of your harvesting. So that it means I think every experiment have a very good reason, and then if you apply this technique, I think it will be sustained for production of our mice in the future. So if no more question, I think it's time for everyone to before we ending this conference, I would like to announce you again that all of presenter will get the certificate and we will send you by email. And of course, for the oral presentation, we have the award for the best oral presentation and also for the one award for poster presentation. The award will announce on October 19. And on the website, you also can come to the website of the ABB conference, and then the announcement will be announced there. And of course, please take you a little bit time to give us the survey in the visual on the virtual slide. We will have the seminar survey. So take your time to give us a survey on the comments. That is very useful for us. And finally, I would like to take a group photo with everyone together. So please give me a smile before we say goodbye. Okay. Yeah, perfect. Wait a moment, please. Okay, maybe it's okay to see your smile. Even we do like a Zoom meeting, but that will be nice to see everyone. And finally, I would like to give my sincere thanks to our team and our partner, especially SPI, the University, and also all of you give a very informative information for our meeting today. And we were pleased to thank you, all of you again, to have an outstanding conference meeting. And I think next year, if we have a chance, we can meet each other again. So please preparing the time and come to see us again next year. So today, I think it's the time to say thank you very much for everyone. And of course, for more information, you can visit our website again, www.abbconference.kpi.ku.ac.th. Every information will be there. Even your award also will announce there also. So thank you for today again, and have a good time. Thank you. ขอบคุณค่ะ Bye bye. Give you a big hand.
Thank you.
Thank you.
Thank you very much.
Very much.
Thank you very much to all the speakers and moderators for sharing knowledge and your expertise. And of course, also thank you to the audience for your participation. Before we end the session today, I would like to invite everyone to visit the virtual marketplace. This will be where our exhibitors, including some of the world's leading brands, showcase their latest technologies, innovations, solutions, and services in renewable energy, energy efficiency, environmental protection, electric vehicles, and smart pumps and valves. Only these 3 days, 14 to 16 of October, that you will be able to contact the exhibitor by using chat and call function that we provided, or even set the meeting with them. Thank you once again, and we look forward to seeing you next year during 14 to 16 of September 2022 at the new venue, Queen Sirikit National Convention Center, Bangkok, Thailand. Please stay safe and healthy, everyone. Goodbye. ขอบคุณค่ะ
ปัจจุบันเนี่ยขนาดโรงงานที่เราทำเรียกว่า Pilot Plant เนี่ยมีกำลังผลิตประมาณ30เมกะวัตต์อาวแล้วก็สามารถสเกลอัพขึ้นไปถึงระดับ100เมกะวัตต์อาวนะครับเรามี มีความมั่นใจครับว่าถ้าตลาดมันพัฒนาขึ้นไปตามเทคโนโลยีที่มีเนี่ยเราจะสามารถเข้าถึงการผลิตไม่ว่าจะเป็น30เมกะวัตต์ในปัจจุบัน100เมกะวัตต์และรวมไปถึงกิกะวัตต์ที่เรากำลังจะมาถึงครับเราพร้อมแล้วที่จะนำ Product นี้ไปสู่มือของลูกค้าที่จะนำประยุกต์แบตเตอรี่ของเราไปใช้งานในโลกพลังงานแห่งอนาคตที่เราเชื่อมั่น
Installing and maintaining a PV system is generally a safe, secure process. But like the cars we drive, some PV systems have more safety features than others. To protect your technicians and installers, SolarEdge employs some of the world's strictest safety standards. Our integrated safe DC technology automatically de-energizes DC cables to a touch-safe 1 volt whenever the inverter is turned off. This complies with the strictest international standards protecting PV professionals, firefighters, and assets. Inverter-embedded arc detection, installed in hundreds of thousands of monitored systems all over the world, accurately identifies specified arcs and shuts down the inverter to prevent fires. Our optional auto reconnect feature allows for continued safe production until the issue is resolved. And safety meets convenience with our module-level monitoring and remote troubleshooting, which eliminate diagnostic work at dangerous heights. Best of all, SolarEdge safety is built into the system. No additional components or installation is required. Safe DC, arc prevention, module-level monitoring, and secure battery operation. That's how SolarEdge protects your team and your customers' investment.
We are seeing growing momentum around the world among governments, private business, and individuals towards carbon neutrality and net zero. We all want to leave a cleaner planet for our future generations, and hence we must act now. SCG is strongly committed towards sustainability. Our EV solution platform will drive and accelerate the speedier conversion towards smart and clean mobility.
EV Solution Platform is a one-stop service under the Smart Clean Mobility concept that allows partners to run their business worry-free at an affordable cost. The platform covers 3 areas: focusing on finished product with battery safety as the priority. Local production, for example, EV bus semi-knockdown unit assembly line and electric vehicle conversion are manufactured domestically. Maintenance and spare parts service. Charging station. Logistics operation service. And IT platform development for a seamless connection between service and solution, making users more convenient and confident when transitioning into electric vehicles. Customized business models from rental service, sales package, to logistics provider service for products or employees. with Vehicle Incident Insurance Type 1 and Battery Product Liability Insurance. Our one-stop service creates an EV ecosystem that offers worry-free solutions at an affordable cost for our partners. It is also environmentally friendly as electric vehicles emit less carbon dioxide and PM 2.5, resulting in a better quality of life.
I would like to invite all our partners and stakeholders to join us in this exciting journey. We're passionate towards delivering smart, clean mobility to our customers. We will do this, we will make this happen by engaging with all of you in a spirit of trust. And most importantly, we will grow together.
SCG International, trusted international business partner.
Electricity never ceases to flow. Electricity is everywhere. Providing reliable electrical energy for our country is the main mission of the Electricity Generating Authority of Thailand, or EGAT. No matter how high or how deep we have to go, EGAT will bring electricity for the sake of the country and the Thai people. Under the vision, Innovate Power Solutions for a Better Life, EGAT generates electricity with reasonable fuel management. This creates a balance between stability, price, and environmental care. Additionally, EGAT purchases electricity from independent power producers, small power producers, and from neighbouring countries. Electricity is transmitted via the EGAT power transmission grid that stretches across the country to electricity distributors to meet the demand of each sector. EGAT also sells electricity to neighbouring countries to create power system stability for the region. EGAT aims to increase the organization's capability through research and innovative development of electricity generation. To support future needs, electricity generation is conducted with a hybrid system. In addition, the Battery Energy Storage System, or BESS, that supports grid modernization also allows EGAT to access the international renewable energy market. The National Control Centre, or NCC, and the Renewable Energy Control Centre help manage renewable energy effectively. Security of the electricity system is prioritised for both the present and future needs of renewable energy, in accordance with the government's policy and power development plan. EGAT uses its organisational assets to conduct electrical industry businesses. and undertakes investments that help strengthen the power system. EGAT Innovation Holding Company Limited was established to efficiently conduct business relating to electrical innovation. EGAT helps create a high-quality society by sharing knowledge with the community. For a sustainable community. Walks side by side with the Thai people. And protects the environment with consciousness and technology. EGAT operates under good governance principles and core values. The night sky is charming, yet it emphasizes the importance of electricity. EGAT promises that Thailand's electricity system will always be secure, stable, and reliable. EGAT has driven itself into a century of innovation, generating sustainable and eco-friendly electricity. EGAT, power for Thai happiness.
Electricity does not only refer to light from a bulb, but it also implies to hope, happiness, and good quality of life, brightening up all hearts of customers. Therefore, the emerging of electricity generation in Thailand has evidently changed the people's lifestyles. In 1884 to 1960, initial stage of power supply in Thailand and PEA establishment. During the reign of King Chulalongkorn, King Rama V, electricity was introduced to Thailand in 1884 by Field Marshal Japraya Surasak Montri Cherm Sang Chuto when holding the title of Jamun Waiwaranat. Provincial Electricity Affairs was inaugurated after the establishment of Electricity Section under Public Health Department, the Ministry of Interior. A power plant was later constructed in Nakhon Pathom municipality and started its operation in 1930, followed by the establishment of Provincial Electricity Organization. The power supply was expanded to provincial areas and it stimulated local development. More and more power plants were constructed in districts and provinces. Recognizing the necessity of electricity expansion and good governance, the government established Provincial Electricity Authority, PEA, on 28 September 1960. From 1960 to 1980, construction of power plants in provincial areas. At first, PEA's headquarters office was initially located on Rama 6 Road alongside Samsen Irrigation Canal and then moved to Ngam Wong Wan Road in Bang Khen District, Phra Nakhon Province. The inauguration was held on 28 September 1971. Regarding the infrastructure development in Thailand, PEA had developed electricity generation systems under the National Economic and Social Development Plan 1 and 2. The generation system was an uphill mission at that time. PEA encountered considerable difficulties, including the expenses of machine installation and operation, the electrical distribution to various locations, the procurement of electric generators, the mechanic recruitment for installation, as well as the transportation of machines, electricity utility poles and electrical wires to some rural areas at which physical labor and small vehicle were necessary. The rapid growth of electricity demand became the PEA's motivation in transmitting electricity to every household. In order to improve living conditions of people throughout the country, As a result, PEA formulated a master plan for power supply development in provincial areas. In responding to the increasing electricity demand, this has continuously encouraged development of electric power transmission to Thailand's remote areas. In 1981 to 1990, electricity system quality improvement to enhance business and industrial development. Power supply became the significant part of infrastructure and had an important role for the national progress in all aspects. Thus, PEA implemented the electrical distribution system to enhance the generating capability and to support electricity expansion. Diesel engine power plants were closed due to high cost but low efficiency. Electric current was instead transmitted from Electricity Generating Authority of Thailand, EGAT. As for the people living on the island, PEA laid down submarine cables for the first time in Thailand. From Konom electricity substation to Got Samui, people there had enough energy to use since then. Furthermore, PEA began energy collecting from renewable resources, including hydropower and solar energy, to generate and transmit electricity to remote locations. Some energy was sent to the PEA's commercial operation to strengthen the stability of power system. Additionally, PEA employed advanced technology and efficient machinery to improve the operational system in order to offer excellent service and satisfy all customers. In 1991 to 2000, application of high technology to reach international service standard. PEA applied high technology to raise the power transmission and distribution standards. In responding to the growing demand in businesses and industrial sectors, PEA improved power supply efficiency with a better distribution system of 115 kilovolt power station. Modern technology was employed to manage electricity distribution system. Supervisory control and data acquisition, SCADA, was adopted to automatically control electricity distribution. PEA was eventually able to carry out maintenance without power failure to increase efficiency of power distribution to industrial sector. In 2001 to 2010, organization development to international standard in energy business. The rapid progress of science and technology was the deciding factor to determine the direction of structure and mission development, as well as personnel potential development and the resource management for maximum efficiency. PEA built in every field of work in order to form learning society and foster persistent development of organization, getting ready for the international level with our modern technology for high efficiency of electric power distribution and our good service. The ISO 9002 guideline was also applied to the whole organization for quality assurance. In 2011 to 2020, a journey to digital utility. Regarding the concept of customer-centric services, PEA provides a positive customer experience with easy and quick service everywhere. The organization has changed some CSR business strategies to CSV, creating shared value and operating to satisfy stakeholders' demands in order to become a sustainable business. Aiming for an excellent organization in the electric power industry, PEA improves efficiency of every working system, fulfills expectations of customers, and manages the organization according to good governance principles. Move forward with human resource, digital technology, and innovation. Let's create shared value to society and environment with digital technology and go on a journey to digital utility. PEA.